Okay, so let's continue with chapter 7, and we have some more vocabulary terms here. So, decontamination is more of a general term. It kind of means, using a physical process like scrubbing, and then trying to get rid of as many microbes as possible. It's related to disinfection and antisepsis, excuse me. So, sanitization is any cleansing technique that requires scrubbing, and that's what's meant by mechanically removing microorganisms. Sanitizer, you guys know what that is. You use hand sanitizer all the time. Also, soap, any type of detergent fits into that category. So, sanitary means that it's safe for normal use, but not sterile. Okay, so think of our drinking water. I hate to be the bearer of bad news, but yep, your drinking water has what we call fecal coliform, which is basically poop bacteria in it. But they are at such low levels that our body is not going to become infected by them. So, yeah, our drinking water is not sterile, but it's considered sanitary. And really, anything that you eat or drink is going to be sanitary, but not sterile. Okay, if you wanted to eat sterile food, you'd have to eat astronaut food that would have no flavor in it. So, yeah, you're always going to be eating microbes, but they're going to be at safe levels. Or remember, most microbes don't cause disease anyway, so it doesn't matter. Degermation. So this is a term that means reducing the numbers of microbes on human skin. And basically, another way you can think of it is short-term antisepsis. So remember, antisepsis is using a chemical to try to get rid of as many microbes as possible on living tissue. And it can include things like a pre-surgical betadine or chlorhexidine scrub. Degermation is basically doing that, but just for a very short time. So perfect example is using an alcohol wipe right before you give a patient an injection. You just need that antisepsis to be there for a very short time. So that's why degermation basically means short-term antisepsis. Sanitation is a term that refers to public health measures. So things like having a water treatment plant or having a system where our garbage gets picked up. Those all fit into the category of sanitation. And whether we're talking about water treatment, etc., it means that, yeah, there's going to be microbes present, but they are going to be monitored and maintained at a level below which would make a person with a normal immune system sick. So if you ever hear about boil orders on the news, notice the news normally says if you are immunocompromised, you should be, definitely sure to boil your water because, highly immunocompromised people don't have a good immune system, and they are less likely to be able to fight off levels of microbes that were pathogenic that would not make you sick, but they could make someone who's really immunocompromised sick. Okay, but that's how sanitation guidelines are planned. It's always, people with a normal immune system. What are the levels that won't make them sick? So when we talk about things where we're deciding, what chemical or process should we use to get rid of microbes? So we need to know, does it require sterilization? Obviously you're not going to use sterilization in a patient's room when the patient is discharged because you can't put a room in an autoclave. So you use disinfection. But if it's small things like surgical tools that are going to be used to cut into patient tissues, absolutely, those are going to require sterilization. So is it something that we're going to reuse again and again? Or is it something that we're going to use once and throw away? Can it withstand chemicals, the heat and pressure of an autoclave? Will it penetrate the material to the necessary extent? And really, is it cost and labor efficient? I mean, autoclaving hospital rooms is not cost and labor efficient, but using a disinfectant solution to do that is cost and labor efficient, and it's safe for the patient who is going to be using that room. So those are the things that go into picking the correct method and the correct chemical. So what happens to microbes when they die? Well, we've already talked about the bacterial growth curve and how once death starts to happen in the death phase, it happens exponentially. So when you reach a certain level, what we call, basically kind of the therapeutic level of whatever chemical you're using, bacteria are going to start to die, and they're going to die exponentially or logarithmically. So death will happen when you can either damage the cell membrane or some other structure and cause the cell to kind of disintegrate, or if the cell is being prevented from reproducing. Maybe you do something with its DNA, so now it can no longer undergo replication. That is also going to result in microbial death. So here are a couple of charts that I want to show you, and trust me, these are more common sense than anything, but basically on the y-axis, so here it's talking about the number of cells. So we're going from a small number up to a large number. And then on the x-axis, we're talking about how much time. So it'd be going from 0 minutes up to, whatever. So, yep, a lot of these are just common sense. So for example, in this one, the more cells you have, the longer it takes to sterilize the material. Same thing here. If you have a low load of bacteria, it's going to take less time to sterilize as opposed to a high load, which means much more bacteria. And then over here, it's much easier to kill vegetative cells, meaning just regular bacteria and fungi, than it is to kill spores, or also like protozoan cysts, because they have that hard outer coating. So yep, takes a lot longer to kill them than it does just regular cells. And over here, this is what I'm talking about when I mention cidal and static. So a microbicidal chemical is going to kill everything. It's going to cause death, and death is going to happen logarithmically. When we're talking about microstatic, it's going to hold the bacteria or whatever at that level, and it's going to prevent any new growth, but it doesn't kill everything that's there. Okay, so that's what a microbistatic level is. And most of the time, microbistatic levels are used in drugs that the patient is going to take inside their own body. And the reason for that is to try to reduce side effects. So other things that can influence how well these chemicals work, first of all, how much is there? Is there a lot of bacteria? Are there few bacteria? What type of microorganism it is? What is the temperature and pH of the environment where you're trying to kill them? What about the concentration of the chemical you're using? Is it better? A lot of chemicals work better actually when they're diluted as opposed to really concentrated? What is the mode of action? Is it ripping apart the cell membrane? Is it doing something at the level of the DNA? How is it actually killing the microorganism? And then finally, is there stuff there, kind of gunk, that's interfering? Okay, so like if you're trying to disinfect your shower and you have a bunch of soap scum that's on there just left over from soap, it's going to take more applications, and it's going to take longer to get rid of the actual microorganisms, simply because you have, soap scum could be considered interfering organic matter, and it could also inhibit the action of the chemical that you're trying to use. So here are some modes of action, and modes of action just means how are the chemicals working. So sometimes We've talked about this with penicillin, right, which is a drug, but you can affect the cell wall, either block its synthesis. Maybe there is some type of chemical like bleach that actually digests the cell wall. And what that does is if the cell only has a cell membrane to protect it, it can become very fragile and it's lysed very easily. We've talked about this, how when you remove the cell wall, you can basically pop the cell like a big balloon. Also, there are chemicals, and this is basically how your laundry detergent works. They belong to a class of compounds called surfactants. And what surfactants do, I believe it's shown right here. So these are the surfactant molecules, the blue right here. And what they do is they actually push themselves down into, or the fancy word is intercalate, into the lipid bilayer of the cell membrane, and they cause the membrane to disintegrate. So that's how your laundry detergent works and your dishwashing detergent. They are all surfactants. And so they will destroy the cell membrane basically just by kind of ripping it apart. There are also chemicals that can be pretty nasty that affect the cell at the level of either transcription, translation, or replication. So there are some chemicals that will bind to ribosomes to stop translation. There are some that will bind irreversibly to DNA, which is going to prevent not only replication, but also transcription. You're not going to be able to make any messenger RNA if you've got a big chemical that is binding to the DNA. But you have to be careful when you're working with these types of chemicals because they can be what we call mutagens, which means that, if you happen to get them, if you're not wearing proper PPE, they can get into your DNA and cause pretty bad mutations. And we've already learned about how mutations can potentially give rise to diseases like cancer. So they are chemicals that you want to be very careful with. Okay, so I'm going to go ahead and stop here and we will pick up here with lecture number 3. Okay, thank you.